very excited to have Dr. Mark Davis and Dr. Vincent van Unen from Stanford University with us today to discuss their Crohn's research that PROPEL A CURE is proudly funding. Dr. Davis is a world-renowned immunologist and is the director of the Stanford Institute of Immunology, Transplantation, and Infection, a professor of microbiology and immunology, and a Howard Hughes Medical Institute investigator. He is well known for identifying many T-cell receptor genes, illuminating how these cells recognize diverse antigens. His laboratory pioneered studies of the biochemistry, genetics, and cell biology of these molecules and T lymphocytes generally, which play a key role in orchestrating immune responses. Currently, he is focused on a systems-level understanding of the human immune system, and interestingly, for Crohn's patients, his work has concluded that the variation in most immune system parameters are not driven by inherited variation, but rather by environmental factors. And Dr. Vincent van Unen, an award-winning postdoctoral researcher from the Netherlands and Rubicon Fellow at Stanford's School of Medicine, is working in the labs of both Dr. Mark Davis and Calvin Koo, using the most advanced technologies, including organoids, to unravel the mysteries of intestinal disease. His goal is to figure out what the environmental factors are, specifically antigens, putting a name to them that trigger IBD. So joining us today are a couple of our board members, Al Fliss and Angela Clark and Ildiko Mayes, as well as Lisa Kelly. And thank you all for joining us here. I'm really happy to see you all. So my first question is, can you share a little bit about organoids? And so what are organoids and why did you decide to pursue this path?
Vincent, you want to take that, or do you want to direct it at one person?
I'm happy to give my perspective on this. First of all, thank you for the kind introduction, Annabelle. It is a pleasure to be here. So my background in my PhD program in Leiden, I investigated a lot the immune cell composition in the intestine of patients with chronic inflammatory diseases, such as Crohn's disease and celiac disease. And based on that research, we identified that there were many specific immune subsets in the intestine to Crohn's disease that are different from celiac disease and that are different from a healthy intestine. So we are having an increasingly awareness of all the types of immune cells that are aberrant in the intestine of patients with Crohn's disease. Now, in order to understand how these immune cells function and through which mechanisms they may contribute to the inflammation these patients are experiencing, we need methods to basically investigate their functionality. And therefore, we were very interested in working with Calvin Koo and his organoid system, because organoids are basically a 3D culture system of growing mini organs. So from a biopsy of a patient with Crohn's disease in the lab, we can regrow mini organs of the patient in a dish. And we have been doing this already for 2 years successfully. And what is very interesting about these organoids in Calvin Koo's lab is that they do not only contain the epithelial layer of the intestine that is responsible for absorbing the nutrients. They developed as the first people, the first researchers, I should say, a way to make organoids that they also maintain the intestinal immune system. So the fact that we can grow a mini gut from a Crohn's disease patient that still contains the immune subsets that are likely responsible for the inflammation gives us an opportunity to study them in the lab, because then we can alter them, we can trigger them, we can remove them to really figure out what are the interactions of these immune cells that result into inflammation, in the hope to get a better mechanistic understanding of the disease.
Does anybody have any questions to ask Vincent specifically?
I just wanted to ask how many patients were involved in this cultivation or this initial stage with the organoids or the tissue that was produced.
Right. So we have 2 years of experiences growing them for celiac disease. And that is where we really established the model, especially postdoc Antonio Santos in Calvin Kuo's lab has really driven this research in setting up this organoid system. So all the characterization of it has already been well established in that setting. Therefore, we could quite easily implement it for IBD patients. And this is now ongoing work. And so far, we have grown organoids from tens, 40, 50 patients so far. And a large body of that research has been devoted in getting them to grow well, characterize them, at what time should we be looking at when, as the organoids grow, do they still reflect what's going on at the tissue level, and where is that window of opportunity? So there's a lot of characterization that we first needed to do. Now we have a good understanding of that. So now we are continuing in trying to figure out what is really going on with the disease.
Okay.
If I could just elaborate on that in my perspective. If for many decades, the dominant paradigm in human translational research was a mouse model of disease, and there are many mouse models of IBD, and yet they have not really produced anything of value with respect to human disease, and actually at least the clinicians I've talked to say they don't really model any of the IBD types, including Crohn's, very convincingly. And yet this was kind of like what everyone was focused on. And what happens is then these ideas take on a life of their own, and they become like the dominant model, whether they work or not. And about 15 years ago, I was, wondering why human immunology wasn't going anywhere. And in fact, people were abandoning it because it wasn't going anywhere. But they would just refocus on something else in mice. And I just thought that this is kind of crazy. And if we don't know enough about the human disease, and the dominant model is not informing us, when is a model not a model, as Lewis Carroll might have said, and that is when it's not predictive, and yet again, these things sort of have a momentum of their own and funding, and they have leaders of the field promoting these things. And so I thought, well, this is not working, and we should be doing something else. And what we should be doing is looking directly at patients and analyzing patients with these actual disease, and not depending on these models, as convenient as they have been and are, because it's just in a mouse. We have to really get busy and figure out how to do this. And then you run into the thing, well, you can look at a lot of things. You can characterize diseases, and we're getting much better at characterizing diseases in patients. We've done a lot of that, and healthy people as well, because you don't know what the disease really is until you know what health is, and health has quite a broad range. But then you get to a situation, well, there are lots of things that you would like to do in patients, but that are illegal or immoral, and you can't do them. They would arrest you if you try to do this. So you needed some way to dig deep into the disease, and organoids have been the way to do that. Is if we can if we can mimic the disease, again, it's like, okay, we had this mouse model, forget it, let's have an organoid model, because then if in fact is even better than a mouse in terms of being able to dissect the components, because if you have it in a dish, it's just so much easier to work with and you can get so much more information.
There's been a lot of research regarding the genetic susceptibility to diseases like Crohn's, but there is also acknowledgement that environment plays a bigger role. And so can you explain how your research will clarify the environmental factors? Will you be able to tell if it is a pathogen or if it's food additives or even specific foods, for example? And will you be able to put a specific names to these triggers?
I think what the organoids, if it's successful as it's very promising, what Vincent has done so far, is you can really look at the pathogenesis and the triggers for disease. Not going to be able to look at diet or the microbiome particularly, but those are things you can do in patients in terms of actual patient studies, you know, that can change their diet or analyze their microbiome. And those things are going on and will go on and are going to be part of a picture. What the organoids can tell us is, will this drug work with this patient?
So I'm curious, Professor Davis, would you then see this potentially down the road as being a way to personalize treatment, meaning you would have a newly diagnosed patient and each new patient would have their own organoid model developed at some point?
Absolutely.
Is that what you envision?
Yeah, absolutely. Right. And that's also something that Vincent is going to be working on, I think, pretty soon. And I know it's something Calvin's totally ready for. It's really the ultimate personalized medicine, you know, as applied to these intestinal diseases. But just maybe on the role of these genetic screens, they've been very popular because the technology has been well developed to look at large populations for genetic tags that could predict disease or correlate with the disease. But the results have not been very impressive because and these are hugely expensive and time-consuming studies. And you know, in a perfect universe, then you might have found, and I think the original hope was you'll find one or two genes that were absolutely critical for Crohn's or for inflammatory bowel ulcerative colitis. But unfortunately, biology is not so easy, and there are hundreds of very, very tiny genetic effects associated with the diseases. And at the end of the day, even like the first big paper on like the 35,000 patient cohort, identifying 150 loci that were correlated with inflammatory bowel diseases, they only account for 10 to 25% of the risk, meaning that's just
that's not the end game here. Yeah.
No, well, to me, it says this is the wrong experiment. You know,
right,
yes,
right.
Yes, you spend a lot of money. Yes, it occupied the, you know, many academics, but nah, not working. And it's the same actually in multiple other or multiple autoimmune diseases as well. Diabetes, MS, it's been the same story, that these things are not easily traceable to one particular or even two or three particular genetic defects, but that you know, there's a lot of ground, and at the end of the day, there must be a much more important environmental factors involved in this. You know, if in identical twins or classic studies of type 1 diabetes or MS, where if one twin gets, say, MS, there's only a 30% chance that the other twin would have it. And these are people that have lived together for probably the first 20 years of their lives. So if there's even some of that, they're eating the same food and getting same micro exposure. So that was, again, in research, we're only interested in things we don't understand, and we sometimes it's like going to Vegas. It's like, hey, well, maybe this will work, and you try it, and unfortunately, unlike Vegas, where right away whether it works or not, in the research business, it can be decades before hey, this didn't work, and we got to try something else.
Yeah, agreed. And I don't think we really understand any of the triggers here. I mean, honestly, except for maybe a mild genetic predisposition linked to, like you said, numerous genes out there that really don't ultimately get you anywhere. Because so that's what makes, I think, the particular research you're doing so exciting, because again, somebody's actually looking at the triggers. Because again, I mean, I think the bulk of the disease, I mean, and again, I've had Crohn's disease since I was 19 years old, and I've been through the plethora of inhibitors, and immune blockades and things like that. And ultimately, I see it as a band-aid kind of, ultimately, trying to affect an outcome, but not actually dealing with the trigger again, because ultimately, if you hit the trigger, I mean, if you know what the trigger is and you could, modify the trigger or block the trigger, then the sequelae down the road are essentially not there anymore. Yeah, sure.
I'm curious to know whether in your organoids from all of these, tens of patients, whether you see heterogeneity. We know it's a very heterogeneous disease and presentation.
Yeah.
Do you see that translated in the organoids themselves?
I think it's early, but Vincent just gave a talk about what he sees, and yeah, it's tremendous variation. And sometimes that can disguise a more simple, something that could be simple. I mean, I maybe I'm dreaming here, but sometimes the multiple different manifestations could just be a result of different times from the initiation of the disease and the course of the disease and who knows what else. But yeah, it's definitely coming up in what Vincent is doing in terms of the characterization of the organoids. And we'll just have to, we just have to get more data and see, do we see unifying trends, or do we really have 10 different diseases here? Hopefully not, but yeah, it's too early to know, other than, yes, at least we're definitely seeing a diversity, and whether that could be collapsed down with specific threads, we don't know. It's too early to tell, but also partly that's where also the drugs will be valuable, because drugs that work, that the same drug working across a swath of patients that we have an organoid characterization of will be helpful, and we'll see what the drug's effects are in the organoid, and then is that a huge diversity, or is that a small diversity? So those are all things we could learn, I think, really quickly. I mean, I think this is going to be, I mean, especially as we emerge from the pandemic, I think we're going to be able to know some stuff very quickly. And that'll be one thing. Again, their tools, they do work, at least for some period of time with some patients.
So do you see to explain to the people who'll be watching this video, how will your research get us closer to a cure?
By knowing the pathway, by knowing the triggers, absolutely. That's something we hope we can, and Vincent is trying some things now. But even knowing the trigger, what is being triggered, what cells are being triggered? How are they being triggered? Why are healthy people's, the equivalent healthy people's intestinal organoids not triggering, or are they triggered? And how did this aberrant pathway get going? Can you trace it back to some sort of origin, or guess what the origin might be, and then do further work to try to see if that's correct? And if it is correct, then could you reconstruct just what went on? So for example, and that's part of what encourages me a lot in terms of our work, is that we have identified a whole new pathway to suppress autoreactivity, especially of T cells and maybe other cells as well. So, and we see that this is active in multiple autoimmune diseases. Everyone we've looked at so far, actually. And the job of these cells seems to be to kill autoreactive T cells. So, but obviously it's not, this pathway isn't working as efficiently as it should in people with active disease, but we can see it's working well in healthy people, and particularly in the context of infection. And it's sort of, I think, revealing a link between infection and the rise of self-reactive cells in an infection, even in healthy people, and how that is normally controlled. So since this seems to work in most people, why is it not working in people with autoimmunity? What went wrong? And also that what gives us hope that we could reverse it and cure the disease is most people are not born with autoimmunity.
Right, exactly.
That they had a perfectly functioning, working immune system, and then suddenly they didn't.
Yeah.
And targeting this particular tissue and not other tissues typically. So that suggests there's an aberration there. It might be about mutation. It might be about some sort of failure in the repression process. And then if we knew what that was, that could then be targeted. Right now, people didn't even know about this pathway a year or two ago. In fact, we're still trying to publish a major piece of the story. So, but I think it immediately becomes a question of, let's look carefully at of what's happening. And now we have tools. We have these really terrific tools, both genetic tools and gene expression tools and other kinds of tools that can allow us to zero in on the population that should be controlling, or there are multiple cell types that are involved in this. And we need to look at each one and say, how does this differ in autoimmune situations in patients than others? So that's we have this whole new tool set, and we're putting it to work here in addition to the organoids. But the organoids will be part of it, because we should be able to see, or at least work backwards from triggers to, what is being triggered and why are these normal control mechanisms not doing their job. But probably what's happening is they're doing some of their job, they're doing most of the job, but not quite enough. And so you have these periodic swings. And that's something we've seen even in animal models of autoimmunity, is that there's a yin and a yang sort of thing going on. And that also gives hope that, okay, we're just at, there's an imbalance here. It's not all or nothing in most cases. There's a dynamic, and if you're sick, it's because you're on the losing end of this dynamic. But it's a normal dynamic at least to some extent. But then why is it going wrong? Why are you losing control of this? Partially because, again, you're not experiencing the full disease 24 hours a day. It seems to be sometimes you are and sometimes you're not. So that's a clue. I mean, we're detectives here, and we're kind of looking for clues that could help us go in the right direction and place the right bets in terms of what else we should be looking at. So it's still I think a lot of the clues will be in the patients, but we'll be able to really get down in great detail with the organoids. So it's a continuum. It's not one or the other, but it's all these things can give us a different view.
I know that you've been involved in the COVID-19 research, and has that helped you inform your research about, Crohn?
Absolutely. Yeah, because we were finding all these things, this pathway in autoimmune diseases involving this whole type of T cell that killed other the bad T cells, and yet we also saw it in healthy normal people, but at a lower level. It was elevated in many autoimmune patients, but it was a very low level, like 1 or 2% of the CD8 T cells of healthy people have this, and the gene expression looked, quite similar. There wasn't any real big differences there from autoimmune patients. So the question was, so what is the normal role of this? And from other work, we hypothesized that infection triggers autoreactive cells. And in fact, a lot of people with autoimmunity cite an infection that happened just before they develop symptoms of autoimmunity. But of course, most people are infected all the time and don't develop autoimmunity. And so, we were thinking that, and there's old literature that said that infection breaks tolerance, which what that means is that you have autoreactive cells, but they're held down. They're held in a quiescent state unless there's an infection. But infection brings them up if they react with the pathogen. Most don't react with the pathogen, so that's okay. But there are some that are going to react with the pathogen, but they also react with your gut or your kidneys or pancreas or whatever, and those are gonna be activated. And we've also seen in some other studies we've done, we've also seen evidence of this two-tiered system where these self-reactive cells stay down unless there's an infection-like circumstance. So then COVID came along and we thought, well, okay, if this is true, then we should see these cells elevated in COVID patients. And we do, big time. And actually correlates with disease severity. So that just fit perfectly with this idea that this is a normal mechanism and normally built to control autoreactivity in the context of an infection. We've also seen in flu-infected patients, and they don't get autoimmunity. So COVID is special in terms of its ability to promote autoimmunity in severe illness and death in a way that flu typically doesn't. But it's a part of this, the same mechanism is going on. And we think this is a major finding in terms of the relationship. What is this pathway doing normally? And then somehow something is not working in autoimmunity.
So I assume that you're not able to really, perhaps you can, because it is highly unpredictable what the results are going to be, but do you have any idea when we'll see meaningful results from Vincent's work? Or
I think this year is going to be a good one. What do you think, Vincent?
I think so too. I think we did a lot of the hard but necessary work of characterizing the system and make sure that what we're looking at is something that's useful for the purpose we have in mind to encapsulate the Crohn's disease in a dish. And all that has been figured out, and now we are moving into the more exciting part. Now that we know what we're working with, how can we really utilize this to study the mechanisms in Crohn's disease? And once we had a handle of that, we definitely want to move towards, can these organoids be used to guide therapeutic choices, but also can it be used as a platform to discover new therapies? And I think to elaborate a bit on what Professor Davis said earlier about the yin yang, that really resonated with me too, because especially in the intestine, I think we have to also realize there are many cells in there that keep our gut healthy. Especially in healthy people, the majority of the immune cells do an excellent job in maintaining tolerance. And don't be, fooled by the current drugs being targeted, because they're not so targeted, in fact. And I think that is what really needs to become more clear, is what exactly goes on in Crohn's disease. As Mark was alluding to, mechanistically, we need to be able to pinpoint exactly what is driving the inflammation. For example, TNF is produced by almost every immune cell I looked at, and the majority of those immune cells will keep your gut healthy. TNF also helps epithelial cells stay healthy. So definitely there is going to be other side of the coin of taking that treatment, and now it is all balancing out the bad from the beneficial effects, and therefore we really need to have more handles on what is exactly going on, and are those autoreactive T cells that we can specifically eliminate. Is it a certain immune cell talking to another immune cell that is driving the inflammation? Are there specific triggers that put on the entire inflammation system? All those types of questions, I think our current research efforts is trying to shed light on, to get more targeted insight that could both help in getting the right drugs to the right patient. It could also help in developing novel therapies that are in fact targeted to eliminate the bad side of the intestinal inflammation, while not affecting the cells in your body that keep you healthy. And I think that is the big challenge that we're trying to take on. And with the tools at hand, I think we can now really look at the disease in completely new light, in all the expertise in Professor Davis' lab and the technology developed and Calvin Kuo. I think we really have a unique momentum to look at this disease more specifically and using humans and not mice.
Well, I thank you so much for helping us, the supporters, particularly who are going to watch this, understand the significance of your research. The viewers, I would like to say to our viewers that if you'd like to contribute to this very, very important and promising work, please donate, because as you have heard, it has an enormous amount of promise. Please remember that 100% of your donation will go to this work. It will not go to us. There's no overhead, as we are all volunteers and we work out of our home. So I really appreciate the time that you've taken, both of you, and the board members here, and for this video to put it together so that we can put this online and on our website and on Facebook, and to share it, because I think it's very exciting and hopeful. And as we know, hope is really, really important for all of us. And so we really are quite grateful for your work. So thank you very much. And thank you for being here today.
It's our pleasure. And thanks everyone on the board and Annabelle for all their support. And it's really inspiring for us as well. So
thanks.
Thank you both.
Thank you for your work.
Absolutely.
Thanks.
Thanks.